You already know the basic equation for photosynthesis, but at A level, we throw away the simple input-output model. You are going to look inside the leaf at a molecular level. We will track the exact flow of energy, electrons, and protons across membranes and see how inorganic carbon is fixed into the organic molecules that sustain almost all life on Earth. You will also evaluate practical investigations, like using redox indicators to measure the Hill reaction.
We begin with the physical machinery. You will explore how the ultrastructure of the chloroplast is perfectly adapted for its job. You need to know exactly where the primary and accessory pigments sit and how they absorb specific wavelengths of light to kickstart the whole process. Examiners love testing your ability to identify these structures from electron micrographs.
Once light is absorbed, the biochemistry begins. This is where light energy is captured. You will trace the path of an excited electron from a split water molecule down an electron transport chain. You will learn how cyclic and non-cyclic photophosphorylation use chemiosmosis to generate the ATP and reduced NADP required for the next phase. This is a sequence of redox reactions. If you understand the flow of electrons and protons, you can answer any structured question the examiner throws at you.
Next, we move into the stroma. This is the Calvin cycle. You will see how the enzyme Rubisco fixes carbon dioxide and how the ATP and reduced NADP you just made are used to reduce that carbon into triose phosphate. This is the crucial turning point where a gas becomes the building block for carbohydrates, lipids, and amino acids. You must memorize the acronyms and know exactly how many turns of the cycle are needed to build a single glucose molecule.
Finally, we scale up to the environment. You will analyze how light intensity, carbon dioxide, and temperature act as limiting factors, controlling the overall rate of photosynthesis. We will also look at C4 plants like maize. When examiners want to separate the A-star candidates from the rest, they target the C4 pathway because it demands a synthesis of biochemistry, leaf anatomy, and ecological adaptation.
By the end of this unit, you will be able to map complex biochemical pathways directly onto chloroplast structure. You can trace electron flow, detail the exact steps of carbon fixation, and analyze the environmental factors that dictate crop yields. Get your flashcards ready for the acronyms and never study the biochemistry in isolation from the cell structure.